Do you hear me well? Or yeah, good enough. All right. So thank you everyone for being present today here. And I'm not a computer scientist at all. I'm a physicist, actually, a medical physicist. But I wanted to, I guess, instigate you to see the techniques that we use and see how they basically come out from computer science and how you can also contribute. You can also develop and do this kind of thing. So yeah, I'm Bruno. I will be presenting prognostic modeling of cognitive decline with confidence quantification to you today. And yeah, let's get started. So a bit of context. This is not computer science at all. We know that cognitive impairment happens with age, neurodegenerative diseases like Alzheimer's, vascular dementia, or other disorders. And we know from the literature and from the clinical experience that the latest marker of decline is shown here in blue. It's the MRI marker. Basically, brain atrophy happens before you develop. The green curve shows the cognitive impairment, so the evolution from normal cognition to mild cognitive impairment to dementia. And this atrophy is a marker of neurodegeneration. So you get synaptic losses, you get cell death, and that happens in normal aging, and also it's accentuated by disorders. And well, how do these markers of atrophy appear in an MRI? So here I have 8 examples of MRIs, and one of different subjects, one for each decade of life. And you can see that some markers are quite clear. You get these, the ventricles. These are cerebrospinal fluid compartments inside the brain. They are very much enlarged the older you are, for example. This means that the surrounding brain has shrinked. You get atrophy, basically. But it's not only that. You also see some of these, the sulcal depth here, they are increased. So the brain is basically shrinking with age. And this is, it's not always the case that you are losing neurons, but it's also the architecture of the cortex that's changing. All right, so if we want to study that, we have to quantify this thing. How can we quantify atrophy, or at least the morphometry, the morphology of the brain? So we have these possible markers here. In pink, we have, for example, cortical thickness. We have the cortical gray matter, and we can measure the thickness. We can measure that manually, for example. That was how it was done 20, 30 years ago. Or you can also do that automatically. And you can also measure other things like the surface area of the cortex. On the other hand, if we want to study the correlates of cognitive impairment, we also have to quantify cognitive impairment. And one instrument that allows us to do that is the Clinical Dementia Rating. It's not really used in the clinics, but it's very commonly used in research. So you might have seen the current, the new clinical trials for treatment for Alzheimer's, for like the lecanemab or aducanumab. They were based on the Clinical Dementia Rating. It's basically a questionnaire. It has to be answered by an informant that spends most of the week with the patient. And you have 6 the rows here. Oh, sorry. Yeah, you have the rows. There are 6 domains, and they're called box scores. And you can summarize. There's multiple summaries, but one way of summarizing this is what's called the sum of boxes. So you just add all the numbers. And the ratings go from no impairment, 0, to severe impairment, 3 per box score. So the most severe level of the sum of boxes is 18, is when you have 3 for each of these. And this is late-stage dementia. All right, so we know that neurodegeneration and cognitive impairment, they are linked in aging and in these processes, pathological processes. But then the real question, can we use these individual patterns of atrophy to predict? So we have the data in the present, and we want to know how it's going to look like in the future. Can we predict the future cognitive impairment to give us better, more precise prognosis, or improve clinical trials, improve studies, do, for example, personalized treatment? And to answer that question, we got data, a lot of data. We have data from almost 4,000 participants from these 2 cohorts, the ADNI and the OASIS. EDNI is tailored for Alzheimer's research. In OASIS is broader, has less people with dementia actually. And, and we have MRIs from all these people, and they are longitudinal, so we have multiple MRIs for most of them, and we have a collection of more than 10,000 brain MRIs here. And we also have a collection of almost 20,000 CDR sessions from the same subjects. And from the brain MRIs, we go and extract these cortical morphometry features. And yeah, we measure cortical thickness, we measure hippocampal volume, we measure gray matter, total volume, other morphometry features using FreeSurfer, which is an open source tool that does that for us automatically. And these plots also come from FreeSurfer. So we have more than 500 features from brain MRI. And we also have, obviously, some risk factors which are important, like sex, age, education. APOE, you might have heard about it because last month was the Alzheimer's Awareness Month, so it was very commonly seen in the media. APOE is a gene, and it's a big risk factor for dementia. But then we have the last feature, which is time. So we also include the time between the MRI session and a CDR session in the future. That's, I would say, the main feature here. So we are going to model this thing with a machine learning model continuously in time. So given the MRI session today, what's the expected CDR in the future every single point in time? And then we can compare it to the sessions that we actually had in the training data. And to do that, we are using, I'm very sure most of you have this context, I don't have to say it, but gradient boosted decision trees, which is an ensemble method where you train sequentially, trees sequentially, and each subsequent trees try to predict, they're optimized to predict these pseudo targets, HI here, and they are the gradient of the loss with regard to the target. So each subsequent tree should make the loss go a little bit lower, and we use LightGBM for that. It's I would say this second or first most common gradient boosted decision trees library today. And why not use a deep neural network directly from the image? Well, we could have done that, and we might do that in the future. It's just that from tabular data, usually this is the first thing you should try. That's what people do in competitions. They do it in research as well. Gradient boosting tends to work very well. And then we separate EDNI into training and validation set based on subjects, because we don't want to have images from the same subject in both the training and the validation set. That's leakage, you can never do that. So we have a 20% split, and then we have 4 domains here. The first is what we call FreeSurfer to CDR, given a FreeSurfer estimate what's the CDR in multiple times, points of time in the future. Then we should also compare to something that's simpler, because an MRI is an expensive procedure. It costs a few hundred francs here in Switzerland. So we do a simpler test, which is CDR to CDR. Acquiring the CDR takes 30 minutes. It's very, very fast. So then we take the CDR in the present and try to predict CDR in the future. And then we combine both. We have 2 sessions, one from FreeSurfer, one from CDR, and we try to predict CDR in the future. And obviously the easiest, which would, if we only had the risk factors, just so we have a baseline to compare. And then we use OASIS as an external validation set, so it has no overlap with EDNI with regards to subjects or scanners. It's completely independent, and we validate these 2 domains. So, all right, what's the performance of our models? We have the 4 domains here. In blue, we have the FreeSurfer to CDR performance. These are windowed correlations, so it's a 12-month window. And we see that we have 0.7 around that of performance. That's the correlation between the predicted and the true CDR SOB score, which, well, the number itself doesn't mean anything, right? But it sounds like a good number for a correlation. And then we can compare to the covariate model. So yeah, the FreeSurfer really adds more to that. It really improves performance much more than just having the risk factors, which is usually what the clinical trials have to begin with. They recruit people based on the risk factors only. But the CDR to CDR model, the red one, In shorter time windows, it's actually better than having the free surfer features, the MRI features. But that makes sense because CDR SOB does not really decline so fast. So it has an autocorrelation. And because of that, if you know my CDR score today, I am very sure you can guess what it's going to be tomorrow, or perhaps 1 year in the future, but perhaps not 10 years in the future. And the purple one is the combination of the two domains, and we can see that it's actually the optimal all the way through, and that's what we would expect. So if you deploy your model, an example would look like this. We have the risk factors here for this subject, and you have their brain MRI. You preprocess that with FreeSurfer, get their morphometry estimates, and then imagine you want to recruit people that, you have your clinical trial, you want to recruit people that are going to display cognitive impairment, because if you recruit people that will not, they will not help you in the clinical trial at all, because you give them the treatment or placebo and their CDR is 0 everywhere. So they are really lowering the power of your clinical trial. And then we apply the model to this subject. We have the true CDR SOB values as these black dots, and the predictions are the red line. And yeah, for this cherry-picked or hand-picked example, it works very well. It gets very good correlation with the actual scores. But then you deploy to another person that you never saw. It's a brain that you've never seen something like that before. And well, now you're deploying the model. You don't know the true CDR SOB scores in the future. So you get predictions, but can you trust the predictions? Like the brain is very, very different, and it's actually not even a brain. This is a bell pepper I got from the internet, but you cannot trust predictions. The model always gives you predictions. So how can you make a way, construct a way of obtaining the confidence of the model? There are multiple ways of doing that. I will present you just one. You can, there's also approach based on quantile regression or other things. But here I wanted to introduce probabilistic predictions. So instead of predicting the number, the CDR SOB, as a continuous number, we predict the probability of each level of CDR SOB. So from 0, 0.5, up to 18. There's a problem. The prevalence of these levels is very low in the training data. The high ones, for multiple reasons, some reasons are also regarding the actual studies. But yes, for many of the levels, we don't have data at all, or very few data. The colors are in logarithm scale, so the dark purple is basically one observation. But we know that the CDR SOB comes from the box scores, the 6 rows in that table. And these are less sparse because it's only 4 or 5 levels. And it's also nice because we could then disambiguate the different contributions to the different domains of CDR, because they are very different. So how would it look like? So imagine you have the predictions for each of the box scores. They are shown here. So each row sums to 1 because it has to have 100% probability. And how can you translate from this to the CDR SOB probability mass function? This is basically the convolution theorem. It tells you that the probability density function of 2 random variables is given by the convolution of their PDFs, their densities. And then that's all nice and well. We do that now. We predict the 6 box scores, and then we can deploy the model again. So the same example as before, it would look like this instead. So in the background, you have the probabilities for each of the levels across time. And I choose to put this purple line here to show you that you can still get a single number if you wanted. That's the expectation. You could also compute the median or the mode, the most likely level, for example. And the dashed purple lines are the 95 percentile confidence interval, sorry. And you can see that the probabilistic expectation does not really match well with the true values, which are shown in light blue. But the confidence interval really covers the data. And we can measure these across time, and we get a good coverage for the true CDR SOBs. And it's also nice that the uncertainty of the model increases with time, or the confidence is lessened. And just to make sure that we do not get a huge performance decrease because of that, we go and show here the correlations from the expectation just as before, there's some degradation here. It could have been accounted, but we are losing some here to get something else in return as well. And I also go and apply, so yeah, the colors do not match, but here I have the free surfer and the covariate model in ADNI. And here in purple is the free surfer model in OASIS, and the green one is the covariate model in OASIS. And you see that the generalizability is not optimal. You really lose performance when moving from ADNI to OASIS. But also remember that OASIS has less people with high levels of dementia. So the variability in OASIS is much lesser. The range of cognitive impairment is much lesser. And that attenuates the correlations. But if you would compare with regards to mean absolute error, for example, OASIS has like very, very low error. So I would claim that the model does generalize well, at least to the normal subjects in OASIS. All right, so we have probabilities, we have the expectation, and we can evaluate the expectations here. But how can we evaluate the uncertainty, the confidence? So one way to evaluate the confidence is to do a calibration plot. This is one way of seeing that, and I'll explain you what it is. I know that it sounds complicated. In the x-axis, we have the time since the MRI session, and in the y-axis, we have the probability that the model outputted of having CDR SOB equals 0, or in other words, the probability of not having cognitive impairment. And this is all in validation data. Left is ADNI and right is OASIS. So if the model says, and I obviously thresholded here, I binned the time and the probabilities, but if the model says that the probability of not having cognitive impairment is 100%, when you take subjects from the validation set, you would expect that if the model is calibrated, in reality, these people that you said had 100% of not having cognitive impairment, they should not really have it, because you said 100%, right? So then we go and we compute the proportion of people that do not have decline in each of these bins, and that's the color. So you can see already that in ADNI here, we said 100%, but really the proportion was less, more like 75%. And in OASIS, we said 100% for these times, this point in time, and it really looks like it's 100%, so that's good. But then I can actually plot the difference from the predictions to the proportions. And in red, it's everything that's red is places where we overestimated this probability. So we said 100, but it was actually less. We were overconfident. And in blue, in OASIS, you can actually see there's also some areas where we are underconfident. We said the chance was, the probability was smaller than it actually was. And the nice thing about these calibration plots is that you can actually use them as a model, because if you would take, this is just an example, I didn't do this for this presentation, but it's in our study. So if you say that, all right, so in ADNI, this should have been lesser, 0.7, well, you can just add this information to the model, and then when you say 100, actually you output 75%, for example. And this is just a way of calibrating probabilities. We also validate the model with regards to biological reality, I would say. We can measure feature importances for gradient-boosted trees. And here I plot the importances. I add the importances for any feature that comes from the same region. That happens for these cortical regions, for example. But yeah, the exam date, the T, is the time difference from the MRI to the CDR session. And it's the most important feature. It makes a lot of sense because the data covers more than 20 years. But the most important feature from the brain is the left hippocampus. And every feature from the brain I colored with as a percentage of the left hippocampus importance. And then I can plot these things to the brain itself. And we see in this upper right figure that the most important features were the hippocampus from both hemispheres and the parahippocampal gyri. And this really matches with what we would expect, because the hippocampus is implicated in memory processes, memory encoding, working memory, And also from Breck staging, which is the pattern of expansion of Alzheimer's, we know that it starts around this area, so the entorhinal cortex, areas that are close to the hippocampus. So yeah, the model makes sense, basically. That's what I would like to conclude from this figure. So a practical significance example then. I had mentioned clinical trials before. So imagine you are recruiting subjects from OASIS before they actually enrolled, and you want to do a clinical trial for Alzheimer's, for cognitive impairment. And well, as I said before, most of them do not show decline at all. They start at 0, so you are only recruiting people that do not have dementia, and they stay at 0. Like 4 out of 5 stay at 0. They never show decline up to 6 years in the future. So if you recruit these people, they will make your statistical power in your clinical trial lesser, because you give them placebo or treatment, and the fact is that they will keep the 0 because the treatment will not help them. They would not have decline to begin with. But then we can separate these people using our model, and we put to the left the ones that have less than 50% chance of of presenting decline at any point in time, and to the right we put the ones that have more than 50% chance of exhibiting decline any point in time. And in the right one, you see that we have a much smaller subgroup because of that. It's only 77 health participants, but among themselves, 6 out of 10 exhibit decline. So if you would recruit from this population and then do the subsequent analysis, subsequent studies from them, you increase the power of your clinical trial. You can also do it the other way around, and then you can make the clinical trial more economical. You can also lessen patient burden, because bringing patients is a burden. They sometimes cannot really be available all the time. Or you can make your clinical trial better, because if you recruit 500 subjects instead, you can get them from this subpopulation. So you get much increased power. And I would like to talk about obvious limitations and future opportunities, which I think are the place where the computer scientists can really contribute to this area of research, either in academia or in industry itself. We choose the features in this study at this point in time. So yeah, we measure cortical thickness and other things, but that's a limiting factor. It could have been done in a different way automatically, I would say, with representation learning based on deep neural networks, for example. The morphometry as a marker of neurodegeneration is also limiting. It does not encompass everything. We could look into including more modalities, for example, or doing deep neural networks with mixture of experts, products of experts, and do multimodal deep learning. Obviously, there's also methodological implications. The CDR-SOB is limited. We should generalize to other instruments for sure. There is a lack of data diversity, and we should extend this generalization to other populations. And we could also model modifiable factors, because these exist and we do not account for them. So changes in lifestyle or treatments or medications that the patients take during their clinical course. And a take-home message, obviously the first one, I would say that morphometry is highly predictive of cognitive decline in longer time windows, so 5 years to 10 years in the future. And we can use probabilistic modeling and probabilistic thinking to get a valid confidence quantification for this domain, but this is really just an example. We could have done this for anything else that's interesting. You can do it for span filtering, you can do that for anything that you find interesting, trading or whatever. And the probabilistic prediction, they give you this adjustable level of confidence, but the thing is that they do not come out of the box perfect. They are obviously prone to error. So probability calibration can make these confidence levels actionable, so that when you say that there's 100% chance of having the case of or detecting an attack or doing a correct trade, you can be sure that it really is 100%. With that, I would like to thank you, and thank you the VIScon organizers for inviting me to this talk. And I also have to acknowledge my colleagues in the University of Zurich, the Swiss National Science Foundation for the funding for this study, and also the patients, the investigators, and everyone involved in ADNI and OASIS, which is a very rich data source. So thank you all.
Well, thank you, Bruno, for this very interesting talk. Now is the time for questions. If you have any, raise your hand. I'll bring the microphone over to you. There is one.
Thank you for your talk. I have a question about this being applied in like real life, I guess. So is this already in use, or what is still needed until this method could be applied to patients and then maybe like used to apply some preliminary treatments or something like this?
Yeah, that's a very good question, because when we're talking about tech, sometimes we lose sight of when this actually is applied to real life. So my model specifically is not applied, so yeah, I have to say that. But this type of research, this type of model is used already. There are companies that do that. There's one, I don't have any affiliation with them, they are called Perceive AI, and they are in Montreal, in Canada. And they do consulting, and they do sell the deployment of their model to big pharma companies that want to make their clinical trials more effective. So it has real life applications already, for sure.
Thank you.
Any other questions? Yes.
Thanks again for your talk. I have a question about the features that you used. There was like something with FreeSurfer, I think.
Yeah.
What exactly is that?
Sorry, I think I really skimmed over these details, but I said FreeSurfer many times. Sorry if people were lacking this context. My fault. So the automatic morphometry estimates from this slide, they are done in a tool called FreeSurfer, which is open source tool for automatic processing of brain MRIs. And yeah, that's where it comes from. That's why I said FreeSurfer many times in the presentation, actually. Does that answer the question?
I think so. Like the features, I think on the slide before.
No.
Those like kind of features you said, like for example, the size of the gyrus or something like that.
Yeah, well, they are shown here. So, for example, we have or you mean like this slide? Sorry, this one.
Yes.
Yeah. So, what's the question again?
If those are those kind of features.
Yeah. So the features that we have for cortical morphometry, we could have more, but they are listed here. The features that we took from cortical regions, because the brain has multiple compartments. I just skimmed this part. But there's the cortical, the neocortex, basically, where you have the cortical gray matter, which is the outer surface of the brain. And because it's a surface, you can really measure, surface metrics, like thickness, so the thickness of the cortex, of the gray matter. And you can do that for each of the regions. And we measure for these regions, so that we have these 148 neocortical regions. They include gyrus and sulci. It will measure volume, area, average thickness, and also the standard deviation of the thickness inside the region. We could measure other things. We could also measure curvature. We could measure the whole surface, so the virtual surface that the brain is encapsulated in. And that's only for the cortical regions. We also have other features. For example, the volume of the hippocampus. The hippocampus it is cortex, but in brain MRI, it does not really look nice. It's very hard to process, so we just give up and just compute the volume because that's easier. Yeah, did I answer your question in the end?
I have another question. I know there are like different brain scan methods. Like, would it be useful to use different ones? Like, would there be a benefit?
Yes, absolutely. So, as I said, we have a very narrow window into atrophy. We are just looking at where each region starts and ends, so that's only morphology. But with MRI or with positron emission tomography, we can also measure other things. We can, for example, measure the content of myelin in the brain using MRI. We can measure the susceptibility tensor with MRI as well. We can measure synaptic density with PET. All these things sound important. The things that T1 weighted, which is the images we usually see shown, which is the one in the example. So this is a T1 weighted image. They are much more common. They can be acquired in 10, 15 minutes, or with current accelerators up to 5 minutes. And they are very easy to get right, because the other ones, they require processing and other stuff. This comes directly from the scanner. But yes, absolutely. Having other modalities should be important. And if you want to do a thesis or something up to that, you could definitely do it.
Okay, thanks a lot.
Thank you. Any other questions? Because otherwise, then I would proceed to thank you again, Bruno. And we also have a small token of our appreciation for your talk.
Thank you very much.
Small remembrance to VIScon. Then also thank you for listening and being interested. Now is lunchtime. Food is served on the F floor, right in front of where the opening was. Also, don't forget to drink. There are fridges everywhere. Just open them and take something. Also, there are baristas over there. They make really good coffee. Go to them, get coffee if you want. And otherwise, have a good time. And thank you again.